This was the last presentation for today. We now have 5 poster presentations. Each of them will take 3 minutes. And what I will do is I will share my screen and show the poster, and then the person who made the poster can start talking and has 3 minutes to present the information. So I will start sharing. I think Paulo, you need to unshare still. Let's see. Okay, you can start.
Yeah, good afternoon. I'm Dr. Harish from Himalayan state of India. So I would like to introduce myself. I'm heading a team of around 10 researchers, and we are doing research in various apiculture, this program, including Bumblebee. Here, we are in the foothill of Himalaya, and we are focused this research on Bombus hemorrhoidalis, which is quite prevalent over here. And we have conducted various studies. This is just a part which I'm going to present. And in those studies, we have attempted the rearing of this Bombus hemorrhoidalis, using it for pollination. And we have found around 17 bee plants on which bumblebees are foraging, and they are dependent on the different seasons, worked out their diseases, etc., etc. So for this presentation, we have just studied some nest architecture during July to September. We have located few nests, and we have come out with certain observations which are given in the poster. I would just like to mention few things. The first one is that the temperature inside was 24.4 to 29.7 degrees Celsius. It was maintained within the nest cavity, and the entrances were one in most of the cases, except at one place we found 2 entrances. And when it comes to the total strength of the population of these bumblebees, so it varied from different seasons in these colonies from 30 to 113. That means the colonies are quite populous also. And Mother Queen was present in all these colonies. We took these colonies and established those in our bumblebee apiary, and made certain other observations, including number of daughter queens. There were 132 daughter queens we observed, which were emerging, and drone strength was in total 55 in these, varied from 2 to 20. Just would like to mention that contrary to earlier observation, we found that drones, they appeared first in our colony. And then we also noted this colony survival. It varied from 18 to 112 days, and we could maintain those colonies up to November in our conditions. We, out of these 132 queens, we further took 65 queens for artificial mating experiments and 20 for this hibernation after mating. So we have found a good mating success. That is about 60% of the queens, they were mated, taking mating time from 2 to 20 minutes. And those with average of 15 plus, we attempted rearing of those. And we had about 40% success in brooding. That means they initiated this wax cell formation, and 2 queens, they give brood, and workers also 2 to 3 queens. That means about out of 10, 30%. So interestingly, this diapause or hibernation is facultative in Bombus hemorrhoidalis. We have earlier also conducted such experiments, and the queens, without undergoing hibernation after mating, they started this another colony formation. So these observations, all these observations, we are taking into consideration, and we are moving step further for commercialization of Bombus ambrialis for use in our polyhouse conditions. That's all. Thank you.
Thank you. Please start with the presentation. Is Douglas Sponsor here? Or?
Yes, I was able to sort out my connection problems. Am I supposed to be seeing my poster now?
Oh, it's on my screen, but it's not. I see that's not on the Vimeo Live at the moment. I'm not sure why.
Okay, I could try to just use very descriptive language.
I will wait. I will try it once more.
Okay. Okay, I see it. If we could just go to full screen. Okay, well, greetings from Germany, though as you can probably tell from my accent, I hail from the land of species poor and mostly yellow bumblebees in the eastern United States. But the study I'm reporting on today is set in the Berchtesgadener Alps of southeastern Germany. And we collected bumblebee wildflower visitation data over 3 years, together with independent floral survey data. So we have quantitative data on the bees, on the flora, and on their interactions. And the point of this study is that when we talk about range shifts in bumblebees, it's not as simple as just sliding bumblebees along an abstract elevation gradient. These are actually mountains. And this follows up very directly on Leon's talk and on Paulo's talk. Mountains, the linear elevation gradients of mountains are punctuated by nonlinear biological responses. And it's important to understand that if we are to understand the effects of climate on bumblebees and flora. So briefly, we modeled using nonlinear techniques the abundance of bumblebees and flora. This first plot on the bottom left. We see many nonlinearities, but the one I want to emphasize is that above 1,500 meters, which marks the tree line ecotone, we see a sharp decrease in the abundance of floral resources. And also the tree line ecotone itself was the distributional interface between high elevation and low elevation bumblebees. And interestingly, the floral taxa that those high elevation bumblebees foraged on were located mainly below the tree line ecotone, largely outside the range of their bumblebee visitors, which may put those bees in a precarious situation with respect to range shifts. When we look at beta diversity, we see that overall beta diversity is dominated by the floral community. So we have a very stable bumblebee community foraging on an extremely dynamic floral community. And that raises interesting evolutionary questions about how bumblebees have evolved to forage efficiently in such an extremely variable floral environment. But I want to zoom in closer on that. This last graph here on the right, we used a technique called generalized dissimilarity modeling to model the rate of beta diversity change along the elevation gradient. And again here, we see that the tree line ecotone at around 1,500 meters in our system strongly punctuates the biological response to elevation in our mountain system. And in both the flora and in the interactions between the bumblebees and the flora, beta diversity increases sharply beginning at the tree line ecotone and continues to turn over rapidly for the remainder of the elevation gradient. So what this tells us is that when we think about the responses of bumblebees and flora to climate, we can't think of it in purely linear and continuous ways. We need to understand that this is punctuated by the nonlinear responses that occur because mountains are ecosystems and not simply abstract elevation gradients. And we should focus perhaps our research efforts on this tree line ecotone where both the most rapid and probably the most ecologically significant changes can be expected to occur. Thank you.
Thank you. I will try to share the next one.
Okay, thank you. Hi, I'm Julia. I'm affiliated to the University of Tübingen, and I want to give you a quick overview about our little Bumblebee project, where we are focusing on two closely related bumblebee species, Bombus terrestris and Bombus lucorum. And as Vincent showed earlier, those two species look morphologically quite similar, and they're also hard to distinguish. And this is the reason why they're grouped together in a lot of studies. And so species-specific knowledge in an ecological sense is rather limited. And the knowledge we have is mainly based on Western European studies. And in areas of their distribution where less research is going on, such as in Eastern Europe, there's still the need to investigate these patterns and maybe also verify known patterns. And this led to our little project in Romania and Bulgaria, where we first of all genetically identified our samples, so we're really sure which sample belongs to which species, and then we modeled their species distribution. And Leon just earlier explained very well how that species distribution modeling is working. So we wanted to disentangle their different habitat preferences and maybe also get to know why they co-occur in some areas and why they also show distinct distribution and patterns in other areas. So for Bombus terrestris, we could verify their known wide distribution, and we could show that they're more abundant in warmer, drier, and lower areas. And for Bombus lucorum, we could show a clear restriction in mountainous areas in our study area, and that they were more abundant in colder, wetter areas with more vegetation. And these results and the clear indication that they have some different habitat requirements and different habitat specializations, we were really interested in finding out if there were some species-specific adaptations that could maybe help explaining these differences in distributions. And for this, we used a landscape genomics approach by using so-called gene-environment associations. And these methods combine genetic markers on the one hand and environmental variables on the other hand. And what we can get out is associations between certain loci and environmental conditions. And here on the poster, I can only show you the results of Bombus terrestris, but we found some associations of certain loci with precipitation variables and cold temperatures. We could identify some ecological meaningful patterns about all our study area. And what is really exciting is that we found loci under selection, which were associated to the exact same environmental variables, also determining the distribution of Bombus terrestris. And this eventually can hint towards local adaptations to certain habitat characteristics Bombus terrestris is living in. And I'm currently working on the Bombus lucorum dataset, and I hope I can soon have some broader conclusions and share these exciting results with you further. Thank you.
Thank you. Then we go on to this one.
Hello. Hi, this is Charlie Jung from Korea, and I am working in Andong National University in Korea. And in Korea, we have about 25 species of bumblebees, but also we are importing one commercialized species, which is Bombus terrestris. Do you hear me okay? And while we are using Bombus terrestris in the greenhouse, and we are facing some of the ecological risk associated with this alien species, so that we began the project of commercializing the native bumblebee species, which is Bombus ignitus. But as we see here in the title, that Bombus ignitus and Bombus ardens are very similar in morphology. So it is very difficult to distinguish the female morphotype when people are working in the field. And also some people working in the rearing unit, they have difficulty to identify or discriminate. Thus we began this project to identify exactly, so that first we have measured the morphological characters and we compared. And we had measured all the leg character traits and morphological traits on the head and antenna and so on. And we found some difference in the morphological trait. For example, had the height and width ratio, and also the Jena width and Jena height ratio, and so on. So that there are some difference in the morphological trait, but that is not quite distinctive. So that in the second step, we have tried to do that in the molecular species discrimination. So that we had developed several primer and have tested with those primer to detect or to discriminate these two species. And finally, we selected 3 primer which can show that species discrimination. So that using this information, and we can actually discriminate these 2 morphologically similar species. And also for the commercialization of this Bombus ignitus, so that we are working also on the rearing practice, rearing test on their most preferred environmental conditions, such as the rearing temperature and also the food what they need. And so we had some success on artificial rearing inside the laboratory and also in the rearing unit. But the problem is that we cannot continue the rearing system, because always in over several generations, then all the population began to degrade. So that we should solve this problem, and then maybe we can make this species commercially available. Okay, that's it. Thank you.
Thank you. And then the last poster presentation.
Hello all together. I'm Rolf Witt. I'm entomologist and freelance biologist. And with this poster, we would like to give an overview of our ongoing work on the protection of a rare bumblebee species in Lower Saxony in Northwest Germany. That started in 2013 with a break of 2 years in 2017, 2018. And the project has been executed by the NABU jointly with experts. And the NABU is a nature conservation society with the largest number of memberships here in Germany. The main goal has been applied work on rare bumblebee species protection on different levels, from inventories, development of local management plans, cooperations with owners of valuable large-scale habitats. Even though bumblebees are one of the most popular insects, there are no governmental programs of support here in Germany, and the knowledge on the biology of rare species is even poor. So another important part is to organize bumblebee seminars. In addition to classical identification methods, the working group of Jorge Gross from the University of Bamberg have developed jointly with us a polytomous identification app. It was developed specifically for students and under consideration of their problems in understanding written expert descriptions. The work is still in progress and the apps can be downloaded. The aim for this year will focus on locating nests of Bombus muscorum and Bombus veteranus. In the coastline here in North Germany, there are existing still the most important metapopulations of these species in Germany. The search shall be performed by means of radio tracking, but also with manual methods in a surrounding often difficult to access. Our aim is a resettlement in different prepared greenland and primary habitats like wheaties with Erica tetralix. Important is a careful and gentle treatment of the limited bumblebee donor populations. So we want to avoid catches of emerging queens for the resettlement. Our strategy is to relocate a colony in a nesting box that will firstly be observed in an initial habitat. Later, the nesting box shall be resettled in the new habitat where the colony cycle shall be complete. There's some experience of the pioneer Eberhard von Hagen, meanwhile over 80 years old, who kept, for example, Bombus distinguendus or Bombus humilis in boxes over years. but there are no experience with Bombus muscorum or veteranus. So before resettlement took place in remote habitats, we have to do some research work on the nesting biology. We are very interested in exchange of experience and opinions. Many thanks for attention, and big thanks to the organizers of this very great conference. I'm looking forward for the session next week. Have a nice day and bye.
Yes, thank you. So let's see which screen I should go to. Stop sharing. Okay, I think we're at the end of today's meeting. I would like to say a thank you to all the people presenting. I looked at the screen at the remarks people are putting in the chat, and I think everybody really enjoyed the presentations, and the quality was quite high, and everybody was on time with the presentation, which is also very important. I would like to say that the presentations will be online later on, so it will be possible to have another look at them. Next week we will continue this meeting. It's still possible to invite others to join, so it's possible for more people to join next week. And the last thing I would like to say is thank you to Linde Slikboer. Maybe we should have her on screen for a small moment, because she is the one organizing this whole symposium and getting everything done. So there's Linde. So thank you from all of us, I think.
Thank you, everyone. Thank you for attending, and thank you for the speakers as well. Were very interesting presentations, and I'm so glad to host this. It was very nice to see researchers from all over the world. So thanks, everyone, and I hope to see you again next week.
Thanks. We got a lot of information, and I appreciate organizers' efforts and giving us opportunity to be here and a part of the symposium. Good luck.
Thank you.
Thank you. Okay, then see you next week.
Yeah, sure.
Goodbye.